Hello guys, welcome to this lesson. We're going to talk about proteins. But before starting, I want to tell you what we're about to learn. We're going to talk about the definition of proteins, then we're going to talk about the properties of proteins, the organization levels, and the protein denaturations. So let's start. We're going to talk about proteins under functions firstly. Proteins are macromolecules formed by linear amino acid chains. It means that proteins are big molecules which are formed in the form of chains. As there can be many sequences of amino acids, there is a plethora of combinations that form many different proteins. Proteins are synthesized thanks to the process of translation, which is a topic that we will discuss in another video. They can have many functions in the cell. Proteins are in charge of growth and maintenance of tissues. There are other proteins as well that provide cells with rigidity thanks to the fibroid characteristics. For example, collagen. It provides structural support to the body. Collagen is present in your bones, ligaments, tendons, and skin. As well, proteins provide energy to the body, even though not as much as carbohydrates. As well, proteins called enzymes are in charge of causing biochemical reactions. Enzymes can function in many ways. In fact, they can work for energy production, muscle contraction, or digestion. These are some functions that proteins have, but as we have said, they are in charge of many other functions. Proteins contain carbon, hydrogen, oxygen, and nitrogen. Proteins contain as well functional groups, which make them different from one another. There are many types of functional groups. Some may be hydrophilic, others may be basic or acidic, others negative, others positive, or hydrophilic. Proteins are formed by many amino acids. That is why they are complex molecules that have different organization levels. In cells, the proteins are assembled in long polypeptide chains in which the amino acids follow each other as a train wagons, thanks to peptide bonds. Now let's talk about the organization level of proteins. I want to begin making you notice these four structures. Proteins are divided according to their functions as well as according to their structures. There are four protein organization levels that go from the primary structure at your left to the fourth structure, the quaternary structure, at your right. So, I want you to notice that every time the complexity of this structure is bigger from the first one to the fourth. That's why we say that proteins are complex molecules. So, I want to begin talking about the primary structures. They are linear sequences of amino acids. Primary structures are held together by peptide bonds, and they always begin at the N terminus, which refers to the amino terminus, and finish at C terminus, which refers to the carboxyl terminus. These primary structures are strictly precise for each protein. Even a minimal variation can alter or destroy the functionality of the protein itself, making it unusable or harmful to the organism. After you finish this lesson, I highly recommend you reading the article below, which refers to the danger that a not well-produced protein can cause. Once assembled, interactions occur along the polypeptide chain among amino acids, which make them arrange in space, like folding back on themselves. It is a spontaneous process that occurs before proteins fall into tertiary structures. These interactions create simple configurations known as secondary structures. There are two main types of secondary structures: alpha helices and beta sheets. Let's begin talking about the spiral or alpha helix structures. It is a motif in the secondary structure of proteins that is composed of the folding of residues of the polypeptide chain. You can see here that it folds on itself. The shape of this structure is maintained by hydrogen bonds. Since hydrogen bonds break and easily reform, helical structure proteins are elastic. Some examples of such proteins are myosin, which is one of the main components of muscle tissue, and keratin, the protein in the hair. Remember that every protein is composed of a lot of amino acids. The folding of the alpha helices structures occurs between two parts of each amino acid, a hydrogen atom of the amino group and an oxygen atom from the carboxyl group. The binding between these amino acids are not parallel. Therefore, it transforms the polypeptide chain into a helical structure. The other type of protein structures are beta sheets. They consist of polypeptide chains that contain 3 to 10 amino acids. These polypeptides are called beta strands. They are smooth and soft, but not elastic as alpha helix structures. So we can say that many beta strands are necessary to form a beta sheet. Beta strands link together through hydrogen bonding, which is the same process that alpha helix structure is used to link together. It means that the hydrogen atoms from the amino group links to the oxygen atom of the carboxyl group. Beta sheets can be structured into two main types: parallel and antiparallel beta sheets. Parallel sheets, as its name indicates, has all strands directed towards the same direction. As you can see in the image at the right, parallel beta strands link through hydrogen bonds. I want you to notice that the organization of these bonds are not the same in comparison to the antiparallel beta sheets. They follow a bonding pattern that is similar to a zigzag. Then we also have antiparallel beta sheets. These structures have beta strands that alternate their directions. In this case, beta structures are linked through hydrogen bonds that are equally distributed. Now let's talk about tertiary structures of proteins. They are the three-dimensional shapes of proteins, and they are formed due to the amino acid side chains that bond with one another. In fact, a particular tertiary structure depends on the interaction of side chains. Tertiary structures can be formed by one or more secondary structures. Tertiary structures are bound through hydrogen bonds, ionic bonds, hydrophobic interactions, and disulfide bonds. I want to make you notice that disulfide, also known as SS bonds, are linkages of sulfur atoms from different amino acids. They stabilize the folded form of a protein. Lastly, there are proteins that are formed by more than one polypeptide chain. This level of organization corresponds to quaternary structures, which involve the interaction between two or more polypeptides. An example of a protein that possesses this type of structure is hemoglobin, which is present in the blood red cells of mammals and has an oxygen transport function. Here you can see that the quaternary structures are the most complex protein structures. Finally, it should be noted that secondary, tertiary, and quaternary structures depend on the primary structure, which is to say the linear sequence of amino acids. After we have talked about the structures that proteins can have, I want to talk about the process of protein disassembly, which is known as denaturation. It is a process that doesn't occur at random. It occurs due to the external factors like elevated temperatures, extreme of pH, organic solvents, etc. When proteins become denatured, they lose their 3D dimensions of structures, and therefore they are not able to function. In fact, when a protein is denatured, you interfere in many levels of protein structures. Denaturing a protein can affect sometimes one or two levels. For example, when you're cooking an egg, you're going to break the structure of the egg. In fact, all that you are doing when you cook it is that you are heating it, and therefore you are gradually colliding their secondary and tertiary structures. And what you have as a result in the scrambled egg are only primary structures, the primary sequence that is still together. So you unfold these secondary and tertiary structures. Well guys, that's all for this lesson. I hope you like it, and I'll see you in the next one. Bye!